I have the distinct pleasure of introducing our next panel and moderator, so I'll go ahead and do that now. Panel 3 is Accelerated Clinical Trials Adapting to the Pace. Your moderator is Debra Paxton. She directs the Office of Human Research at the George Washington University, which houses and supports the GW IRBs, facilitating the ethical and compliant use of human subjects in research. Ms. Paxton has over 20 years of experience in research as a researcher, research integrity officer, and IRB director. She is proficient in both biomedical and behavior research and serves as consultant to multiple institutions on human subjects and research integrity issues. Deb, the floor is yours.
Great, thank you very much, and hi everyone, and we made it to our last panel, and I think it's a really great one, so I'm very excited to be here with you and share it with you. I am Deb Paxton, and I'm a little bit of a multitasker for this panel because I'm acting as the moderator as well as doing the first talk, which is really, it's going to be an abbreviated talk to kind of set up the rest of the speakers and frame the panel. We're here to talk about clinical trials and COVID and what is happening in the world today of clinical trials and the accelerated pace that we're all experiencing as we strive towards a cure or towards treatment or towards a vaccine. And we've got a great panel. We've got David Demert, who is someone I work very closely with at GW, as he is one of our IRB chairs, and he is a fantastic IRB chair who also just happens to be a researcher who does clinical trials in vaccines. So he's a great panelist for our talk today. And we've got Shauna Browning, who's over at Georgetown, and she's the director of research quality assurance over there. And she's got some great insights about research quality, and then summing it all up and adding the ethical considerations is Sarah Vitone from Georgetown, where she's a clinical bioethicist, and she's got some great detailed insights about the ethics of accelerated pace in clinical research. So without further ado, I'm going to dive right into it. Next slide, please. Okay, so I don't know about you, but we have been experiencing a number of challenges and exciting opportunities due to COVID. It has been a whirlwind of a time, and it's coming out that there are some primary areas of requests that the IRB office at GW is receiving, right? So we're receiving urgent requests for research to accelerate towards a solution, like I said, a cure or vaccine. We also have researchers who want to get in on the ground floor of some of the unique research opportunities that COVID and the quarantine present. And these are across the board in terms of types of research and areas of research, right? It's not just clinical research. We've gotten requests from educators who are interested in the effects of all education moving online, and political scientists who are interested in the effects of different political messaging across the globe regarding COVID, right? So so those are some really unique research opportunities that can tell us, can really contribute to the scientific record. There's also maintenance concerns for ongoing research, right? How to navigate your ongoing research study in the quarantine and with restrictions. And underlying all of these concerns is a heightened awareness and different considerations regarding risk and benefit, right, in the research. So next slide, please. So I'm just going to talk briefly about each one of those. So in acceleration towards a solution, you know, what we've noticed is that COVID is really highlighting for people the flexibilities that are available in the regulations and under various regulatory processes, right? And each one of those we want to be aware of has pros and cons, right? Clearly the gold standard for gaining information and helping get towards a solution is research, right? But because research can, in fact, take some time to get off the ground, some healthcare practitioners are using expanded use options as a way to start treating patients with not yet proven medications. And that's kind of a theme we've heard across this talk, and you'll hear it again here in this panel. We've got flexibility, communication, and kindness, and it's all coming out in this last panel. So I know that Sarah's going to talk a little bit more and in greater detail about expanded use and then clinical research. It's important for there to be prioritization and communication across all stages of the organization at this point, right? So ideally, departments are talking to their researchers, and colleges are talking to their departments, and then colleges are talking to us, right? So that at all levels, there's everyone is understanding what the priorities of the organization are and what the priorities of the research focus are going to be. And what I've absolutely been recommending to those researchers at GW that I'm working with is this notion of incrementation, right? So the idea that you kind of start small and see how it goes, and then maybe grow the research as appropriate, right? And there's a couple reasons for that. Smaller, more succinct or concise research studies are easier to review and faster to review. They're also more flexible and can be more easily adapted to what is being learned in the field. Next slide. And that recommendation holds across the unique research opportunities that COVID is presenting, right? So you, in addition to understanding what you and your organization's research priorities are, also understanding where to start. And that's where I'm targeting in on this parsimony, this notion of parsimony, because one thing we see when there's not a crisis going on is people trying to make or create the biggest research studies they can, right? Absolutely, in the time of big data, there's kind of a thought that there's no such thing as too little data, and when you come into a crisis like this, I think it's really important to kind of pull back and think about what specifically you want to learn, and intentionally collect data, because that will really allow you to consider more specifically and in a better sense the impact of your research on the human subjects, the ethical considerations of your research, and it will allow you to more judiciously direct and use what may be limited resources. Next slide, please. And similarly for maintenance of ongoing research. The most interesting thing here for maintenance of ongoing research are two things: pivoting to accommodate restrictions and evolving restrictions, right? It seems like every week there's some new restrictions on what people can do or how people can interact. And so being aware of those and making sure that you are changing your research to keep research personnel as well as your participants safe is really important. That's going to involve some flexibility and creativity. And it may involve modifying some research questions, right? As we all know, research is you do the best you can with what you got, and it may be that what you got is changing a little bit. So in those cases, you might want to reach out to your sponsors and reconfigure some of the deliverables that you promised them, and absolutely reach out to your IRB office and other regulatory bodies to see what sort of flexibilities can be afforded to you. I think that is one of the best silver linings of this COVID outbreak that I'm experiencing is increased communication with my researchers. And it's lovely. I think we're really developing a stronger professional relationship because there's a greater sense of urgency and collaboration around COVID outbreak. That I'm really thinking is helping us communicate a lot better, and I'm hoping will persist beyond COVID. And one of the things we're doing is helping the researchers reconsider the risks and benefits of their research in the light of COVID, right? So and that's a pretty common piece of guidance then that's been released by IRBs nationally when we're talking about adapting to COVID, is considering the increased risks to people simply from actually coming into a lab or coming in for a visit, and how to manage those risks, how to reconfigure the risk benefit ratio. Okay, next slide. Oops, I got ahead of myself. Sorry, next slide. Thank you. And this is my last slide, and it's the most advicey piece, the most advicey slide that I have, and it's working with regulators, right? So I think throughout most of this talk and throughout the rest of the panel, the things I've just touched on have been expanded and will be fleshed out by the panelists. But I do have a unique perspective that I can offer as a regulator, right? And so there is a lot of guidance being issued by regulators. There's a lot of flexibilities that are being promoted and allowed by regulators, and I think that's great. I'm hoping this will be a learning opportunity for a lot of regulators and researchers to understand and be comfortable with flexibilities. I know that at GW, that is a little bit of a growing pain for some of our researchers and some of our administrators. And I'm really glad to see the federal government taking leadership in the flexibilities that they're affording us. An excellent example of this is the expanded use of convalescent plasma. So this is a kind of, it's an experimental treatment for COVID, in order to allow it to be used, normally, kind of an expanded use needs to be reviewed by an institution's IRB, et cetera, but to cut down on that regulatory burden, the FDA and Mayo very generously is allowing IRBs to rely on the Mayo Clinic IRB for all of those necessary reviews. And additionally, those of you who are in the IRB world or have some experience with single IRB know that single IRB isn't always extra efficient, right? But the other thing that they've done is they've stripped away a lot of the paperwork requirements for single IRB or IRB reliances to really streamline the access to these treatments while maintaining ethical review, right? They did not get rid of it. They're not cutting any corners. They're maintaining that review, and they've then released some of the paperwork burden that would very likely slow down some of these treatments. So I'm advocating for the embrace of flexibility, remembering that at the base of it all are some basic ethical considerations that we always need to meet, and Sarah can talk more about what those are and how we can meet them in a time like this. But don't be afraid to use those flexibilities. And then also when you're working with regulators, here's the two main things that I've learned over the past couple weeks. First, be your advocate. Advocate for yourself, right? Don't hesitate to reach out to us, because we have people reaching out to us sometimes on a daily or more than daily basis, concerned about the timeline of their review and making requests. So, so get in there so that you can demand the attention and the care as well. And when you do, my final piece of advice: be aware of how you're communicating with the regulators. Specificity of information and questions is a really good way to get good answers. So I see some researchers contacting me with kind of very broad help sort of questions, right? Not really asking a question, just kind of stating a problem, and looking to me to provide some solutions. And I am happy to do that, but it's not going to be nearly as effective as the researcher who says, here's a problem I've identified, I'd like to move forward with the following solution, right? And that allows me to give much more concrete answers in a faster timeline. So I recommend that for how you interact with regulators. And that's my experience as an IRB person. I have to imagine that it extends through the federal regulators as well. All right, so I'm going to cut it off now so that we can move on to the other panelists. I'm going to introduce David, Dr. David Demert, who, like I said, is an excellent IRB chair. I'm very happy that I get to work with him, and an experienced vaccine researcher. And he's also a professor at GW in the School of Medicine and Health Sciences in the Department of Medicine and Microbiology, Immunology, and Tropical Medicine. So David, take it away.
Thanks, Deb, very much for that very generous introduction, and it's a pleasure to work with you as well. And good afternoon, everyone. It's a real pleasure that I've been invited to be a panelist this afternoon. I have been asked to say a few words, and I just have a few slides, so hopefully it will go quickly, about DSMB in real time, really in relation to, and I've tailored it completely to be in relation to the COVID-19 pandemic. And I'm gonna, DSMB is in the title, but it really applies to any independent evaluation board, whether that be a safety monitoring committee, say for a phase 1 trial, a data and safety monitoring board, or FDA refers to them as data monitoring committees, DMCs. Really, whatever I'm gonna say applies to all of those different bodies. And I just wanted to start out just to review, point out the most important responsibilities of these boards or committees, and that is to periodically review and evaluate during a study the accumulating safety data, particularly to evaluate participant safety, but also to monitor the progress and conduct of a study, compliance with the protocol, with good clinical practice, which could be majorly impacted by the current pandemic. And then particularly for DSMBs, when we talk about assessment of efficacy. And not only doing these reviews and evaluations at pre-specified time points, sometimes ad hoc, but also to use that review to make recommendations to sponsors, principal investigators, other investigators, and also by extension, most of these findings are also reported to ethical review committees, IRBs. And what are those recommendations? Well, this becomes extremely important in our current pandemic, whether or not a trial should, or individual study subject participation should continue, whether or not a study should be modified during, while it's taking place, or the most extreme example, whether or not a trial should be terminated. And that has become increasingly important in the current pandemic. Next slide, please. So before I get into how the DSMB and other committees might help in study conduct, I wanted to first touch on how DSMBs themselves might be impacted by the pandemic, and that really is in relation to how they can continue to operate. So I just put down some considerations that might kind of come into play in the current response to the pandemic, and that might come to bear on continued functioning of a board. Some DSMB, and this applies to ongoing studies really, that had already been ongoing prior to the start of the restrictions that have come into place in a lot of jurisdictions, at least in this country and internationally as well. But it would also apply to any new study that's going to start, particularly studies that are targeting research on COVID or SARS-CoV-2. That's whether or not members are available. So a lot of DSMB members might have other roles and responsibilities that they might now might become more important. It might be taken away to provide clinical care, for example, or lead up new research initiatives. And then even more logistic things like whether or not the board has adequate teleconferencing capacity. Some boards meet physically. That often can happen now, whether or not teleconferencing can be arranged and so on. Also really important, especially in the current pandemic, is that maybe it's more important, I would say, for phase 1, phase 2 trials where assessment of safety is the predominant concern, is whether or not meetings can be convened rapidly. I've taken as an investigator, been a PI on several phase 1 vaccine trials where very rapid convening of an SMC had to be arranged, and whether or not that can be done given the current restrictions now. If that's not possible with your current board, it might be time to start looking for new members or an entirely new board. Next slide, please. If that is not an issue and your DSMB, DNC, SMC can continue to operate despite the COVID restrictions, how might they help in study conduct? So FDA has just come out, and I think on the next slide I have the reference, FDA has come out with some guidance regarding conduct of clinical trials of investigational products during the pandemic. One of the main things that comes out from this guidance is that sponsors and investigators, PIs, have to assess whether or not it's really feasible to continue an ongoing trial in view of the restrictions and response to COVID. Definitely, since one of the primary responsibilities of the DSMB is to assure participant safety, they can help. So rely on your DSMB, and often, I don't want to say you want to use them as cover, if you will, but often it goes a long way, especially with IRBs and regulatory agencies, if your DSMB makes a recommendation or a safety monitoring committee makes a recommendation that can be put in writing in a document, and then that can be submitted to your ethical review committee. Next slide, please. The document that the FDA actually came out with in about mid-March and then was updated on April 2nd goes through a variety of factors that the DSMB might use to consider whether or not an ongoing trial should be continued or terminated or modified in some way. And primary amongst these is whether or not the restrictions that are in place now in most jurisdictions pose new safety risks to trial participants. So if someone is enrolled, for example, in an investigational drug trial and can't come in to get their next dose of medication, if it's an injection, and so on, how can how do the restrictions affect the risk-benefit ratio, both for an individual participant, but also overall for the entire study? And whether or not the DSMB could help in coming up with potential ways of mitigating those additional risks that might now be present and come into the risk-benefit equation assessment. Even more basic than that is whether or not your investigator or sub-investigators continue to be available. So I was involved as a member of a safety monitoring committee for a trial going on currently in Africa, where the sponsor, based on the DSMB's assessment of the situation and the availability of the investigators and study team, decided to terminate the study because the PI had been pulled in their country to oversee the COVID response. So obviously was not going to have any more availability to oversee safety and participation in the study. You might have your investigators, whether or not other clinical trial, ranging from study nurses, study drivers, if you're doing studies overseas, for example, investigational pharmacists, and so on, whether or not they continue to be available. And not only are the personnel available, but whether or not they have the adequate PPE to conduct potential in-person visits safely, both for their study personnel, but also for the study participants, obviously. Next slide, please. And this is my last slide, just some additional factors that a DSMB might want to consider whether or not making a recommendation to continue or modify a study. Even if potentially you have the personnel and equipment available to do in-person visits safely, your site might have decided otherwise and to have closed. And if that is the situation, and I know there are clinical trial sites where this has occurred, whether or not you can somehow modify your in-person assessment so that they no longer have to occur at the clinical trial site. For example, using telemedicine to conduct some visits virtually. I know part of some of my studies, we've been able to implement this in a quite rapid fashion, or even doing alternative site visits. I have to say, I've never done this before, but I've started doing some of study visits at subjects' houses and homes, if it obviously can be done in a safe way, both for investigators, study team members, and also the study participants. Availability of study product also might become an issue. This personally has become an issue for me. I'm involved in a study vaccine trial that's ongoing in Uganda, and we can't get study product anymore, and we haven't been able to do a shipment there for a couple of weeks, and there's really no end in sight. So being able to supply trial site with investigational product is going to become a factor in whether or not you're going to continue your study, and DSMB could help make those types of assessments. You might have had to, the study team might have had to modify their schedule of visits, or even product administration schedule, and whether or not the preclinical safety and stability data that you have for the study product would allow for that potential prolongation of shelf life, and whether or not study sites have the ability to properly store study product potentially for much longer periods of time than initially planned for. And then finally, you know, what is the logistical capacity of the IT systems on site? Most of our studies here and overseas use electronic data capture systems, whether or not those will continue to function. And there's adequate support at the various study sites for being able to do, to continue that data collection. And that really gets to maybe what my final point will be, is that not only can DSMBs make assessments on safety and whether or not there's a continued justification for continuing a trial, but the other element that comes into play, obviously, is assessment of efficacy for those like phase 3 trials, for example, and whether or not it's feasible and possible to continue collecting the data required to establish efficacy. So your efficacy endpoints, whether if those can no longer be collected, then that obviously also would come into consideration by a DSMB and making a recommendation for study continuation. So I'll end there. There's the FDA guidance that came out is really is excellent, very comprehensive, and gives a lot of good advice and guidance on conducting clinical trials during this difficult time. So I highly recommend taking a look at that. So I will pass it over to Dr. Browning.
Good afternoon. Can everyone hear me?
Yep, we can hear you.
Terrific. All right, so I'm going to talk to you about research quality in the setting of accelerated clinical trials. Next slide. So many of you may recognize this as Haley Carruthers in the London Marathon last year, in April of last year. Almost didn't quite make it to the finish line of the marathon. So I really begin to wonder how many of you might feel like that today, that you're almost right there getting these clinical trials done and you're rushing, but it's a marathon, it's a race, it's a dash, and you're not quite there. I think the most important thing, that there is a race to get to this finish line in accelerated clinical trials, but the problem of quality is when that is not considered, you might not make it to the end where you hope to get to. So although we want to accelerate clinical trials, we need to keep in mind data integrity and quality so that that can be built into this process. By doing that, we can make it to this finish line. Today, I want to talk to you about quality management plans, risk assessment, and quality by design. Next slide. So when clinical research trials need to be accelerated, it does not mean that you have to cut or compromise data integrity and subject safety. It is a fine balance between getting it out the door quickly and managing that subject safety and data quality. It may mean that you need to shift your quality management plan so that you can ensure subject safety and data quality. And it is important to consider where and how this is going to be done most effectively. Next slide. So as we begin to decide how we're going to achieve quality and maintain that data integrity and subject safety, let's consider these key reasons for poor quality so that you can address these issues up front. So many of the reasons for poor quality in clinical trials include inadequate staff training on GCPs and protocol, mostly due to sudden increase or decreases in resources. And some of you may be experiencing that now. You may have staff turnover, you can't replace them because of the cutbacks in employment currently. You may have lots of new studies going up, so you could have this sudden shift. You could also have poor or lack of management or supervision or quality control of task completion during the study. In addition, lack of protocol clarity can lead to poor understanding of what's going to be required during the conduct and implementation of the protocol. And lastly, we need to consider lack of quality control over the collection and recording of the study data. Next slide. So how can we develop a quality management plan where we can mitigate the areas of poor quality that we just discussed? One of the biggest things that a quality management plan or system includes is to define what those quality requirements are. And if we look at site procedures, that can address some of those issues that we just talked about, where there are unclear procedures. This can help make sure that everybody's on the same page and that we all have the same procedures. Forms and templates can help guide study teams to ensure that everybody is collecting the data in a similar and method and making sure that there are areas that are not being missed. Quality control measures could include people double checking data entry, so 2 study team members checking it real time to ensure nothing is missed, a checklist at the end of the visit before a participant leaves or the remote visit ends to make sure you've captured everything with that participant, and then quality assurance. In addition, when we find problems, we can create corrective and preventative action processes to address problem areas and prevent them in the future. So continuous quality improvement activities really support processes that are standardized to help data accuracy, completeness, and data integrity. Next slide. So let's step back for a minute. When does a quality management plan concept really begin? I want to talk about a concept called quality by design. And quality by design is something that is going to take us all the way back to the very beginning when the protocol is being developed. And a quality by design is a system that takes processes such as monitoring and it reconfigures them based on risk assessment. And the purpose of this is to maximize efficiency and coordinate effort. Current monitoring practices may succeed only in detecting those errors that don't have a significant impact on the trial results, where quality by design is really going to look at those key risk indicators. So it is built in from the beginning. It uses a risk management approach, the focus on key risk indicators where the data really matters, such as event rates, number of protocol violations, maybe a query rate, percent of patients with dose reductions. So it's based on those outcome measures or other data that are really key risk indicators for that particular study. An assessment is an ongoing part of trial design, and it begins with recruitment and it follows all the way through the results. And corrective actions are made early. We don't wait until half of the study is done or to the end of the study. Corrective actions are made throughout and early in the study. Next slide. So why is quality by design something important in accelerated clinical trials? So Laundrie in 2012 points out to us that trial quality is defined as the avoidance of errors that matter to decision-making, and monitoring is repositioned as a tool for evaluation and improvement. So if we use quality by design up front, we're using decisions that are going to help us in the long run end up in a better spot. Next slide. So quality by design is defined as quality in clinical trials that is defined as the absence of errors that matter. Not just all errors, but those errors that matter. And those errors that matter are those key risk indicators. So if we look at the left-hand side here, we can start by prospectively examining the objectives of the trial. What are these objectives? And define those critical factors, those key risk indicators to meeting those objectives. And then we're going to focus the effort on those errors that are affected by those key risk indicators. So those are the errors that matter. What is going to make this trial successful in the end? And then down at the bottom, then we're going to take action to prevent those important risk factors. So those key risk indicators. We want to ensure that those are the areas that we're going to prevent those critical factors from negatively impacting the outcomes of the clinical trial. So understanding what data and processes underpin a successful trial is really essential to identifying and managing important and likely risks. And this way we improve quality and outcomes for the clinical trial, do it from the beginning, and we get to that finish line much faster. Next slide. So how does quality by design improve clinical trials? Quality by design really helps the organizations and the study teams prospectively, so up front at the part of protocol design, make us fully aware throughout the trial life cycle of what are the important errors we're concerned about that could jeopardize our abilities to either protect the subjects, so we're going back to that beginning, how are we going to manage this to protect those patients during the clinical trial, yet still obtain efficient, reliable results that are meaningful information. Next slide. So when we look at the quality by design and implementation, it uses this plan, do, check, act. This is a very common method when we're looking at quality assurance. In the plan phase, we build a plan in the very beginning, and it's built into the fiber of the development of the study that focuses on those key risk indicators, what matters the most. And then we move on to do. We implement the study risk management strategies based on those key risk indicators. And then our monitoring is going to focus on those key risk management strategies and those factors that matter the most. So we're going to be leading that monitoring with the indicators of quality and study. And lastly, we don't just leave it there. We're going to actually systematically drive remediation and learning. So based on what we find early after our check, we're going to fix it early on in the clinical trial so that we can learn from what we do and how to fix it so that we can end up with quality data at the end of the study. Next slide. So institutions not just individual study teams, but both institutions and individual study teams can really act. And by acting, we're going to mitigate those risks that will likely lead to errors that matter and determine how to rapidly identify and react when there is an issue. Next slide. So Rory Collins from University of Oxford, I really think this is so important because especially in this COVID-19 environment where we're trying to move forward in the best way that we can in our clinical trials, is that undue emphasis has been placed on data accuracy. We want every little part to be accurate, when in fact, reliable results can be obtained even from imperfect data. So we really need to consider, especially right now, as we re-look at our clinical trials and as we're developing clinical trials for COVID-19 or other research that we want to bring to the table quickly, we need to consider what matters most. What are those key risk indicators? And ensure quality is there at those parts of the study that are really what matters most, and not total perfection, because that's impossible to receive. So all of our efforts, especially when resources are limited, should be focused on what matters most. Next slide. So in the end, we can accelerate not only the finish, so we can get to that finish, and this is a sprint here instead of a marathon, but we can finish it, but on the same side, we have data integrity and subject safety, and it's so important. And by doing quality by design and implementing that quality with key risk indicators and focusing on what matters into the clinical trial, we can really move forward and not forget about these two important factors. So that's it for me. I want to hand it off to Sarah Vitone, and I did provide references for you to have. So Sarah, up to you now.
All right. Thank you, Shauna. I'm really pleased to be here today, and I am a bioethicist over at Georgetown University and work quite a bit in the clinical setting. I also am faculty over there and teach ethics, and it has been my pleasure to be with the Georgetown Howard CTSA for nearly 10 years now, and I work closely with Dr. Jane Otato over at Howard and with the folks at Children's and GW. When advocacy issues come up, I serve as a research participant advocate, and I think that's an important point to make. I spend some time on some of the high-risk studies helping with oversight of informed consent. And in these relatively challenging new times, I think some of those, the work that I have been doing over the long term has some real relevance today. I also want to give a shout out to the number of panelists that have been on the conference thus far, and how the ethics has threaded through most of the, if not all of the presentations today. And I just want to give a shout out for working with such a great team today. The news and the science breaks daily. Sometimes it seems like hourly. And so a lot of uncertainty in the world as we are changing. We have to balance the pace of the knowledge that we have with the pressure to help these patients with the virus, and research is key. We do have experience with moving research along during infectious disease outbreaks. So this is going to give us some grounding, and I'm ready to get started. So next slide, please. Thank you. So I'll be talking a little bit about research ethics, some of the specific things that we see in a public health crisis, and consider some exceptional circumstances and uncertainty. And if I can get to it, we'll talk a little bit about some of the unproven interventions and how we consider those in working with patients and research participants. I also want to give a shout out to all the folks that have been helping to keep our studies going. And that was one of the more unique things that I was involved in as the pandemic brought us to social isolation. The various things that were going on in clinical trials as studies were modified, and where risk to be coming downtown for study visits began to outweigh the issues of not coming. And so I appreciate all the folks that have been involved in that. But I'm going to spend some time talking about research ethics. And the primary paradigm in research ethics is to assure that research participants are protected, and that the research is conducted to serve the participants as well as society. We use a benefits risk approach, but we mainly depend on sound science. And being a bioethicist, it's important to me that the science is good. I don't want participants to be involved in studies where even one lab draw might be not necessary. It's really important that the science is sound. So I spend a lot of time working on reviews and working with research teams. So we depend on and apply the work of the Belmont Report, using beneficence, respect for persons, and justice as our main principles, as usual in research ethics. We apply those using thinking about subject selection, thinking about informed consent, thinking about respect to participants, how we include participants equitably in our clinical trials, that our subject selection is equitable, and thinking about how we apply these principles and ethics in our research helps to build the public trust and holds us accountable. And this is really important, especially in times like this, because we are going to be asking participants or potential participants to be involved in our research. And we may not be able to save individuals, but we are hopeful to create treatments that will save the society at large down the road. And that's a really important idea as we're talking it over with potential patients and participants and their families. I want to also say a shout out to all the regulatory folks that I work with, and I can echo so many people that commented about the FDA guidance being really excellent, and I appreciate being able to work closely with our IRBs to make sure that these issues are handled well. So I'm going to go on to next slide, please. So just to echo our fundamental premise in ethics is that we need ethics to guide decisions and our actions. And we need ethics guiding us more now, not less, in dire circumstances. And they provide a necessary framework, especially in face of what's happening to us today. Next slide. So just to add to the values and ethics that I've already mentioned, when we're in a public health crisis, we also add additional principles and values, specifically addressing public health crisis. And one of those is social solidarity. And I'll just comment, excuse me, that this value refers to the bonds that unify a community. So in solidarity, and we're all in this together, leaders in health and research articulate this idea, and it allows for the public to accept decisions that are made that are fair and equitable and transparent. And transparency in this case means open access to information and decision making, and this then in circle enhance public trust. When clinical trials are going to be provided and there will be opportunity for participants to enroll, we want to make sure that this is made available to the public so that they can feel that they have the trust in us and that we are accountable to them. Accountability is really having a responsibility to promote thoughtful, fair, and consistent recognition and identifying protocols and participants. We need to take care, just as a final thought here, that research doesn't compromise our response to the outbreak and that the provision of appropriate care. So stewardship is something that we also think about, using the right resources. And that in itself is leads into a little bit of proportionality. So I'll just comment there that, and that's really an ethical principle related to justice. Stewardship of scarce resources may include opportunities for research in itself, and how these opportunities can impact outcome is very, very important. Next slide. All right. So these are the general values that we have in research ethics, and it applies also in any kind of emergency research. We want to make sure the science is valid, that the risks are reasonable, and that participants are fairly and selected and have voluntarily provided consent. Consent's might be a little bit tricky here, so I'm going to mention that again later, and that we are protecting the participants to our best ability, and that we are undergoing independent review. I'll just comment that in the idea of consent, we have to have good disclosure and truth-telling. And what's interesting here is that there's a lot of unknown. And I think that that lends itself to that open and honest communication with participants and researchers. And it's important to note that these type of collaborations across institutions and perhaps even around the world focuses on a common good. And this is one of the more important things. So let me go to next slide. As I mentioned, we do have some experience in research in infectious disease outbreaks. So what possibly have we learned and carried forward from our experiences in SARS and MERS, and even more recently with Ebola? And we have some unanswered ethical questions. For instance, what impact would exceptional circumstances have on research? And what about unproven interventions and the goals of interventional research, the goals in terms of the individual versus collective interests? How do adaptive trial designs impact research? And what is the meaning of compassionate use related to unapproved interventions? Next slide, please. And so this is what the news looks like, right? How do we stand still and do nothing? Is doing something better than nothing? And as our keynote speaker commented, how are you going to criticize someone who's trying to help in the face of what we're dealing with as this surge is impacting the critical care? And professionals have a duty to act. So how to weigh this in the face of trying to provide good information and good data to inform the development of some clinical trials. The fear and desperation that we have, and that we are working with researchers and getting information from around the country on how to best put these protocols in place really is important. We have to consider the moral distress of the team and the treating folks, and also the distress of the patients and their families in looking for a solution to this problem. Next slide. So coming out of the Ebola crisis, the question was, do exceptional circumstances change the paradigm? And the Ebola epidemic was exceptional for a number of reasons, including the magnitude of the epidemic, the high lethality of the Ebola virus, its contagiousness, the additional burdens on a fragile health system, and the opportunity to invest a disease that was only available during the epidemic period. And as I read through that particular article that was written in 2018, I thought about that, and I thought, well, goodness, that sounds quite like what we are experiencing, and perhaps we are going to be thinking about this idea of an exceptional circumstance going forward. The concurrence of these multiple circumstances led to the consensus that clinical trials had to move forward without undue delays. And this is important for us to think about now. Next slide. So should we be using randomized controlled trials? Yes. And how do we think about should we be collecting data when we are addressing a single-use protocol, or which might also be compassionate use? The New England Journal of Medicine put out an article just this week around compassionate use, and they had gone through IRB approval for each participant to use a particular drug, which was for secondary use. And I thought it was a great opportunity to highlight a very rigorous protocol. They got information in, and even though it was not a randomized controlled trial, it provided data that could be shared that would then perhaps give information going forward as trials are put together. Next slide, please. So we're dealing with uncertainty, and it's a separate but confounding issue. How we deal with uncertainty over the risks and benefits of an untested intervention is really important, especially when the odds of dying are high and there's no cure that exists. And this leads to some interesting questions. How can we predict risks and benefits? What trial design is best? And in thinking about the ethics, whose opinion should be counted in the appraisal of risks and benefits? Next slide, please. These are some of the areas that I wanted to mention related to PACE. I will mention that exposing research participants to risk is ethically challenging, especially if the study is not designed in a manner to provide usable benefits. It's therefore imperative that all research be conducted in a methodologically rigorous manner, and in clinical trials, the important features of randomization, placebo control, blinding, and masking should be determined on a case-by-case basis. We need to apply the ethics before the research. And that goes back somewhat to Shauna's comment about quality. It's very important to do that up front. We need to develop mechanisms to ensure that accelerated ethics gets review in emergency situations without undermining the substantial protections that ethics review provides. And one option is to authorize advanced review of generic protocols. And I know that some places have been doing this, and I think that that is helpful. I think IRBs are at the ready, and as mentioned already by this panel, have been putting good processes into place to help researchers who are moving forward on a COVID trial. Consent's a little bit tricky, and I will say that its complexity is that when the mortality rate is high or concerning, then patients and their families affected by the virus, they mistakenly believe that being in the research is a saving option for them. And this can become a therapeutic misconception when perhaps it shouldn't be. So we have to really think about how that's done, and that's something that folks in research ethics can potentially help you with. I want to echo the information sharing obligation that is important to these clinical trials during this time to avoid unnecessary duplication of effort or competition among sites. We have to move forward together, and we can do this better together than we can alone. Next slide, please. I'll just make one or two comments here about recruitment and compensation. We're going to be putting together, I know there'll be trials coming up for vaccine research, and we have to think about value and respect and participant selection related to those, as well as access and making sure there's equitable access if resulting treatment does come about. Next slide, please. And my final thoughts were about using unproven interventions, and these are the main characteristics of such. And I'll just say that using emergency use in unproven interventions must include monitoring and documentation and sharing of the results. And I see that we are at the end of our time. So I just will say finally on my last slide that the highlight of our goal is to maximize the contribution that scientifically robust and ethical research can be made to improve the health of people affected by emergencies. Thank you.
Okay, great. Thanks so much, Sarah and David and Shauna. That's how you show up on my screen right now. Thank you so much. I think those were some really great insights. And we've got some time for questions now. As a reminder, please, if you have questions, use the Q&A feature. And it looks like we already have a question. Very exciting. Regarding DSMBs and IRBs reviewing COVID-19 research, how are you reinforcing or correcting for the principle of equitable selection, given that the subject cohorts may be skewed towards a convenient sample? Sarah and David, I bet you have some things to say about that.
Well, I would just say we wouldn't approach it any differently, I would say, than we would any other study. I don't particularly see why we would have a different set of criteria for assessing whether or not a study is going to be equitably selecting their subjects. I mean, we always use that as one of the criteria for approval of all new clinical trials, clinical research. So we always look at the selection criteria that are part of the application, and look at the planned statistical analysis, whether or not there's an adequate sample size, and so on. That's, I would say, that shouldn't change in the current, and maybe becomes even more relevant in the current pandemic. I don't know, Sarah, if you want to add to that.
Yeah, I would say that the questions that I've heard about this are more related to making sure that we're giving, that we're not necessarily looking at necessarily clinical performance. And one of the things that I initially thought of was what the benefit of the CTSA was going to be, because I know that Georgetown, Howard, sometimes we can get a better mix match of participants when we go across. So I'm wondering if that this could be one of the side effects of the CTSA program, that we can work more closely together, because we're already doing that. So I think that keeping ahead of the kinds of participants that we have in our ICUs and how we select those, I don't know, it's just something that I was thinking about there. Appreciate your point, Dave. Okay, great. Shauna, do you have any thoughts to add?
No, I think Sarah and David addressed the question, and definitely collaboration across multi-site institutions and working together is a way to really make sure we're representing populations across the nation.
Great. We have another question. Is ethical review at risk if we constantly have urgent matters, pandemics, etc. that arise? Or rather, how can we be sure that ethical review is absolutely maintained? And I would imagine that she means during an ongoing sense of urgency, right? Well, that's a good question. I'd love to get somebody from an IRB to answer that one. Don't know anyone.
Deb, you can try this one first.
Okay, great, thanks. I think it is an excellent question, and it gets to what I always say, and that what Sarah, you got to a little bit in your talk, which is it is the process that matters, right? There is a process that we follow. A lot of times people think ethics is related to a specific outcome, right? There's only one sort of ethical outcome, but really, in my opinion, ethics are related to the process that is followed to get to an outcome. So as long as we make sure that we are following that process and focusing on the specific requirements of the process, right? So I think that one of the things that we're learning in COVID is that over time, the process has absolutely become sort of inflated if you will, with all sorts of concerns, right? And that's referred to as mission creep when you talk about IRB operations, and it's something that we've been recognizing kind of academically or theoretically as something that can get in the way of an IRB doing its job, but it's really coming home to roost now as an issue, which is that what the IRB does is it adheres to and helps researchers adhere to an ethical process, and that's very well defined. And in times of urgency, it's important to maintain the focus on that specific ethical process, and then maybe realize what other processes that have been inserted into the IRB review process maybe are not necessary. And by that, I mean, there are a lot of administrative processes that get inserted into the IRB review process because of institutional efficiencies, right? But this is a time to say, look, the ethics of the human research here and the protection of the human subjects are really what we have to focus on. So that would be my answer.
Yeah.
Oh, we've got some more questions. Hold on. Okay, how do we prioritize studies across our institutions so that we do not overburden patients with COVID-19 or overburden clinicians who are providing care with multiple studies that want to enroll a limited group of potentially eligible subjects? That is an excellent question.
Yeah.
I have an answer, and then I think, and then David has one too. My answer is going to be quick. It is what I was alluding to or trying to get at when I talked about institutional organizational prioritization. So this is where I think that at the department level or at the college level, where there's a good knowledge of who those clinicians and who those researchers are and who those participant groups are, that those kinds of discussions and prioritizations can happen. And then that would be communicated throughout the tiers of review or administration that needs to happen.
Yeah, that's what I was going to say too. I don't think it's really the role of the IRB to prioritize the different studies that are coming our way. I agree that should happen on an institutional level, but kind of outside of the IRB review process. One thing though I think should be mentioned, and I've already seen this as a chair and IRB member, is that different investigators, sometimes from different departments, are submitting similar studies that probably could be rationalized, and that might be an area where the IRB office or Office of Human Research could try to minimize any duplication within an institution. I'm not sure how to do that between institutions, but definitely within a given institution.
Great. Sarah or Shauna, any thoughts?
No, I agree. This has come up recently at our institution, and it needs to be considered institution-wide, and then bringing investigators together for collaboration whenever possible is really important. And I do agree the IRB might have that level of insight because they're seeing what's there, so they could refer them back to the institution.
Mm-hmm.
Yeah. Okay. I have a question for Dr. Browning. Specifically, how can we better advocate for quality plans for our researchers?
How can we as individuals advocate for it?
Probably, or as maybe as administrators or institutions.
Right. So I think that supporting quality as an institution is an important thing. So if we set up a culture of quality, that's an important piece of doing so. And then from the administration, I think if we have the ability to support at the onset, so one of the things that we're doing is having quality initiation meetings. So at that time where the studies are being developed, we're actually meeting with study teams at that point to help them, hopefully before it goes to the IRB or right at the time that it's being submitted to the IRB if we haven't caught it by then. And then we're really working with them at that point to develop the quality management plan into that. And then setting up the culture again is part of, this is learning. It's that ACT phase. that we're learning. This is not punishment, this is learning. Quality is all about learning and preventing and managing it and helping you end up with the results that you want at the end. So I believe as administrators, it's a cultural mindset that quality is really something that is proactively done, and it is an educational experience, and it's not punitive.